Good afternoon. Thank you for joining us for today's event. My name is Amanda Ramey, and I am the events coordinator for the Department of Chemistry. Before we hear from our speakers, I'm going to walk us through a couple of housekeeping items. Firstly, the chat is available for your comments throughout the event. You can find the chat at the bottom of your screen. Feel free to introduce yourself, share where you're joining from, share words of encouragement for our student speakers, and you can also use the chat to let us know if you need any technical assistance. We should have time for audience questions following each student presentation, although unless it's very urgent, you can just save it till the end because we will have a last Q&A as well. For all of those questions, we ask you to use the Q&A button if you have access to it. This is also at the bottom of your screen, and if you can't see it right now, you can click the More button, which has a symbol of 3 dots, and the Q&A will be one of the menu options listed. Feel free to submit those questions at any time, and we'll get through as many of them as we can. And now it's my pleasure to introduce Jordan Axelson, Director of Undergraduate Studies and Senior Lecturer in the Illinois Department of Chemistry. Jordan will share briefly about the importance of undergraduate research in the department to provide some context for the presentations we are about to hear.
Good afternoon, everyone. As Amanda said, my name is Dr. Jordan Axelson. Although I now serve as a lecturer and director of undergraduate studies for the department, many years ago, I too was an undergraduate chemistry major here at UIUC. I graduated from the specialized chemistry curriculum back in 2010. Consequently, I have an exceptional amount of pride in our department and in the work that our undergraduates contribute toward our research endeavors. As someone who also benefited from scholarships that were generously given by donors, I would like to personally say that I am eternally grateful for those opportunities that those scholarships provided to me. And on behalf of our current students, I would like to extend that thanks to our alumni and to those of you who are here today who have created funds that support those types of opportunities for our students to gain this valuable research experience during their undergraduate career at the University of Illinois. Undergraduate research is an incredibly valuable experience for undergraduates in that it provides chances for them to apply what they learned in the classroom towards solving real world problems, to hone practical laboratory skills, to delve deeper into specific subdisciplines of chemistry, and to gain insight into what chemistry as a career can look like. So again, I thank you all for the gift of experience you grant to our students through your donations. This past summer, we had 28 students sponsored through various chemistry department programs. The 3 main ones that I'll just kind of briefly talk about today are the Chemistry Department Summer Scholars Program, the Merit Program, and the C² program. So students participating in the Summer Scholars Program have already connected with faculty or are actively working in a lab and are thus nominated by their faculty member for sponsorship during the summer. As someone who frequently serves on the committee that reviews this particular group of student nominations, I'm always delighted by the accomplishments that our more established students have already obtained in the lab, and I'm very excited by the high potential that our faculty see in the newer recruits. The summer research opportunities provided by the Merit Program were actually a new addition for us this summer, thanks to a new funding stream. However, the Merit Program actually runs all year long and during the fall and spring semesters organizes additional discussion-like sessions where students can further engage with their lecture courses. To be invited into this program, students must have high academic potential and be committed to excellence. One of our main goals through the Merit Program is to develop a community of scholars among the students. In class, students work together to solve difficult course problems, develop friendships based on their common academic interests, and hopefully inspire each other to maintain that high level of commitment to excellence. Finally, the C² program is predominantly funded by industry partners and also has a strong focus on building community between our chemistry majors and our chemical engineering students. In addition to the summer research opportunities, C² also offers things like peer-to-peer mentorship between PhD students and undergraduates, monthly networking and professional development events, as well as mentorship awards and student travel awards that students can use to participate in national and local scientific conferences. So having said all of that and kind of introduced you to some of the programs that our students have been involved with this summer, I would like to move on to the true focus of this event, which is our student speakers. So I am now pleased to introduce our first speaker, Carlos Nava. Carlos began working in Professor Lisa Olshansky's group back in May 2024, and today he's going to share some of his work on the impact of substrate access channels on protein dynamics.
Thank you for the introduction. My name is Carlos Nava. I'm a rising senior in the specialized chemistry course in the Olshansky group. And I'd like to present the research I've been working on over the summer in where I was investigating the impact of a substrate access channel on protein dynamics. So just a little bit about me. I was born in Arizona and moved to Illinois a few years after that. Once I'm finished with my education, I'd like to go into industry as an R&D scientist. And as Dr. Axelson had said, research really helped me connect the ideas that I've been learning in the classroom and put them into action. And it also went the other way around. So I learned some concepts in the lab first that I may or may not have learned in the classroom, but it really helped me expand really the type of techniques that we could use in chemistry labs. But outside of the lab, I enjoy being outside, just being in nature and generally playing sports. So, to serve as an introduction for my project, in 2011, there was an enzyme known as ZOXF, which was the first of its kind discovered to bind lanthanum. And this was really interesting because previously we thought that the lanthanide series and actinide series didn't have a role in biology. And so we were really curious what role this lanthanum plays, how it interacts with the protein, how it might change biocatalysis, and among other things. The problem was that it was really difficult to study. When we tried to express and purify this protein, it came out in really low yields. And in order for biocatalysis to occur, it needed two helper proteins known as ZOXJ and ZOXG, which help sort of bring out its catalytic features. So our lab developed an artificial metalloprotein, PQQT, to mimic the function of ZOXF so that it would be easier to study and while still retaining the functions that we're interested in. In particular, we noticed that we have an easier time purifying it. It's easier to modify, so we could change certain amino acids to sort of better serve the purposes that we're studying. And it still retains the lanthanum binding that we see in ZOXF. So in order for this biocatalysis to occur, it needs two cofactors, one of which is pyrroloquinoline quinone, known as PQQ, as well as the lanthanum ion. And when both of these are embedded into the center of the protein, known as the active site, that's when it can undergo catalysis. And so our substrate of choice is benzyl alcohol. And when it interacts with our PQQ with both of its cofactors, it can oxidize this alcohol group into an aldehyde group. And so last summer, we actually focused on mutating this protein in order to increase the affinity for the cofactors. So here in our unmodified protein, we found this residue nearby the metal ion, and we mutated it into an aspartic acid, a negatively charged aspartic acid, so that it could stabilize the positively charged metal ion. Additionally, we found this other residue close to the PQQ and mutated it into a tryptophan due to its high aromatic character side residue so that it could interact with PQQ via pi stacking and further stabilize PQQ that way. We found that when we mutated both of these mutations into a single protein, we found that upon introduction of our substrate benzyl alcohol, catalysis had stopped. So we wanted to find out what was going on with our system, why had catalysis stopped, because as you may know, in order to study catalysis, you need it to occur. So a postdoc in our group ran some molecular dynamic simulations to get some insight into what might be going on with our protein. And what he found is that in different environments, with and without PQQ, our protein actually has some structural changes. And the most interesting is when it's bound to PQQ or unbound, it sort of has this closed conformation. However, when it's not bound to PQQ and there's PQQ within its vicinity, it will actually undergo dynamic changes, sort of analogous to trying to trap the protein, or excuse me, if analogous to the protein trying to trap the PQQ molecule. And so upon binding to this PQQ molecule, it undergoes this closed conformation. And we also ran some k-vert analysis to determine if there are any access channels that might be present. And what we found, indicated by the blue spheres, is that there's an access channel spanning from the surface of the protein into the active site, which contains a bottleneck between two residues. And so we hypothesized that if we were able to mutate one of these residues into a shorter amino acid, we would be able to open up this bottleneck and expand the access channel. So it might have, so that our substrate might have an easier time entering the active site, and hopefully that this would reinstate catalysis. And so we got to work. The first thing we did was take our DNA and then mutate the amino acid that we wanted to change, which we could do by PCR. And we monitored the PCR product through an agarose gel, in which we could see the thick bands indicating that our product was formed. And then we could continue growing the protein as normal and purify it through affinity chromatography, which we can visualize through another gel where we have the different washing fractions, and this thick band represents the purified protein. And then additionally, we can utilize the spectroscopic properties of PQQ, and especially its high intensity fluorescent signal. So when PQQ and protein interact together, it actually eliminates the signal, which we can use to monitor the binding. And so once we had our protein purified, we wanted to make sure that our proteins were interacting with the cofactors, just to make sure that the functions were there and we didn't mutate a vital amino acid. So we took a sample of PQQ and titrated protein into it, and we observed that in the presence of excess protein, we had reduced the signal essentially to 0. And this means that our PQQ is binding to the protein as we expect. Additionally, we also wanted to monitor the lanthanum binding to our proteins, which contain the aspartic acid. And we found that there's a change about an isosbestic point up to 1 equivalence. And after that, it shows a different change in the spectrum, indicating that while lanthanum is binding, it's binding non-specifically, which is also what we expect. So once we made sure that our protein was behaving as we intended it to, we wanted to run some kinetic runs in order to see how our protein was interacting with the substrate, and if and essentially how catalysis and essentially monitoring the catalytic activity within our proteins. So here we have 4 different trials with our cofactors and benzyl alcohol. And going in alphabetical order, we have a run without any protein just to serve as a control, as well as with our aspartic acid mutant, which is kind of our baseline and what we use to compare our access channel mutants. Specifically, we can see the product peak grow in at 340 nanometers. And so this is kind of what we were looking out for in our other proteins. When we take this aspartic acid mutant and introduce the open access channel in C, we notice that there's a significant change in the spectrum, indicating that the protein and the substrate is forming a different interaction compared to our run in B. So at the moment, we don't have a definitive answer for what's going on yet, but it's something we would like to explore. And then finally, in graph D, we took our previously uncatalytic mutant, and we introduced the open access channel. And this is what we would hope would restore catalysis. But unfortunately, we just see a general change in the spectrum, which indicates that catalysis had not been reinstated, and it's just a general baseline shift. So just to wrap up, we saw that the opening up the access channel for our mutants doesn't restore catalysis. However, it does impact it for mutations that are able to already have this catalytic activity. And we hypothesize that there's a change occurring between the interactions of our substrate and the protein, which is something we would like to further investigate, as well as run quantitative analysis for the product formation, which we can do by GCMS. And lastly, we would also like to monitor the PQQ binding, which we can assess by stopped flow and observing the rate, so we could compare it with and without the access channel. And with that, I'd like to thank the C-Squared program for funding my summer work, as well as its sponsors, and especially a big thank you to the Olshansky group for being a welcoming and encouraging environment. Thank you. And if anyone has any questions, I'd be happy to answer them.
Yes, and I'll jump back on too. Again, if you have any questions, feel free to type those in the Q&A. I'll do my best to kind of keep track of those. I think real quick, just make sure we stay on our timetable here. I did have one question for you, Carlos. So I know at the beginning you mentioned that one of the benefits of studying an artificial enzyme in this case was that you didn't have to worry about like those helper molecules, helper enzymes to make sure that it's working. Are there any other, I guess, like advantages that you found, maybe like practically speaking, that moving over to this artificial enzyme rather than the original has been like really beneficial to providing insight?
Yeah. So for our artificial metalloprotein, it has really high yields, like much higher than average, which means that we don't have to spend as much time purifying and expressing this protein. And it's also very easy to mutate. So usually we can like run PCR once, and then our mutation will be integrated, and it makes it much easier to work with.
Gotcha. Very good. Very good. Okay, well, that's all I have for now. I might have more questions for you at the end if we have time. But we're going to go ahead and move on to our next speaker. So our second speaker for today is Bolade Abiola-Fagba. She is a rising sophomore who's interested in pursuing a career in pharmaceuticals. So if anyone's looking to recruit, we might have someone here for you. She comes to us from Professor Marianne Aline's lab and will be sharing what she has learned as she has explored the influence of, I hope I'm saying this right, brocosome geometry on nanomaterial durability.
Thank you for the introduction. My name is Bolade Abiola-Fagba. I am a rising sophomore. I am currently undeclared, and this summer I participated in research in the ABC lab. And today my topic would be about exploring the influence of brocosome geometry on nanomechanical durability. And my research is entomology focused, and yeah. So a little bit about me. I was born and raised in Nigeria, but I moved to Chicago, Illinois, where I currently live, and I went to middle school and high school there. I am currently undeclared, but I want to major in molecular and cellular biology, and I chose it because of the courses offered, and I feel like it's a little bit more interdisciplinary. It has a little bit of physics, biology, and chemistry. And my ultimate career goal is to become a medical scientist or work in the pharmaceutical field. Outside of research, I love watching movies and listening to music, and an highlight for the summer is that I watch every movie in the Marvel Cinematic Universe. So introduction. So you might be wondering, what are brocosomes? So brocosomes are nanoparticles found on leaf-hopper insects, and they're known to be super hydrophobic and anti-reflective. And on the picture on the right, you can see 3 different species of leafhopper insects and their respective brocosomes under. And as you can see, they can range in size, and they come in different shapes and sizes, and they can range from about 200 to 600 nanometers in diameter. And so they're known to be super hydrophobic and anti-reflective, and we are open to create like future surface designs that also like contain this quality. And as you can see, in each brocosome, they have these oval faces on them, and they come in hexagonal and pentagonal shapes. So for our research, we hypothesized that brocosomes with a higher percent of hexagons, a higher number of strut rate count, and an higher diameter would be more durable against compressive forces. And so to explore this hypothesis, we collected the leafhoppers at a prairie in Urbana, Illinois, using sweep nets, aspirators, and black light traps. And there's a picture of me at the top left using the aspirator to get the leaf upper insect of the blacklight tribe. And we targeted 3 different species, which are C. insularis, Erythenara fluoride, and Gyponana salsa. To compress the brochozomes, my grad mentor went to Sandia National Laboratory in New Mexico, where he used a scanning electron microscope to the nanoindenter to compress the brochozomes. At the bottom left, you can see a picture of the nanoindenter, and at the bottom right, there's a picture of my grad mentor at Sandia National Laboratory. So while we were doing the compression, there were load force graphs generated like the one on the top of the screen. And using this, we were able to generate the yield force, which is the amount of force a brochosome can stand before completely deforming. And max form, which is the amount of force a brochosome can withstand at a 75% strain rate. And the brochosome stiffness, which is how much a brochosome is resistant to compressive forces. And using ImageJ, which is a image processing software, we're able to quantify the amount of hexagonal and pentagonal faces on the brochosome. We were also able to calculate the amount of strut and the diameter of the brochosomes. And using the data from the ImageJ and the load force graphs, we're able to use RStudio to create box plots graph and determine the statistical significance of each species of brochosomes. So for our results, on the left side, where we compared the yield force among each species, C. insularis brochosome at the highest yield strength, followed by G. salsa and U. fluoride, which means that C. insularis brochosomes can withstand more force before becoming permanently deformed. In the middle here, we compare the max force between each species, and we've also found out that C. insularis brochosomes at the highest compressive strength, followed by G. salsa, then E. fluoride, which means that C. insularis brochosomes are more resistant to like crushing forces at a 75% strain rate. On the right, the graph indicates that C. insularis, comparing the stiffness between each species, the graph indicates that C. insularis brochosomes are the highest stiffness than the E. fluoride and G. salsa brochosomes, which also indicates that C. insularis are more resistant to deformation forces. On the left box plot graph, comparing the amount of percent of hexagonal faces on each brochosomes for each species, we also found that C. insularis brochosomes are the highest percentage of hexagonal faces, followed by E. fluoride, then G. salsa, which indicates that the geometry of the brochosome does play a role in its durability against compressive forces. And the box plot in the middle, comparing the number of struts for each brochosome, we found out that C. insularis brochosomes at a higher number of strut, then G. salsa, then E. fluoride, which indicates that brochosomes with more structural support would have an increased amount of durability. And for the right, as you can see, for the right on the box plots graph comparing the brochosome diameter among each species, we can see that this doesn't follow the pattern where C. insularis normally has the highest amount of data. We found out that G. salsa brochosomes actually add the largest diameter, followed by C. insularis and E. fluoride, which indicates that the size of a brochosome might not be an important factor when it comes to durability against compressive forces. So in conclusion, we found that the C. insularis brochosome, although they didn't have the largest diameter, they had the highest number of strut count and percent of hexagonal faces, as well as a higher amount of yield strength, compressive strength, and stiffness. For E. fluoride brochosomes, we found out that they were the smallest in diameter. However, they had a higher stiffness than G. salsa and the second highest yield strength and compressive strength. And this is most likely because they have a higher more percent of hexagonal faces than G. salsa brochosomes. And for G. salsa brochosomes, we found out that while they were the largest in diameter, they had a lower yield strength and compressive strength than C. insularis. And it's most likely because they have the lowest amount of hexagonal faces, and also the lowest amount of stiffness, which can mean a decrease in like structural support. So in summary, our conclusion supports our hypothesis that broken zones with a higher amount of hexagonal faces and a higher number of strut counts would be more durable. However, the diameter of the broken zone does not in fact relate to its durability against compressive forces. So in the future, we hope to investigate the relationship between other geometric characteristics and broken zone durability, such as the material volume versus the void space volume, which would be the hollow space inside the broken zone. And we also hope to measure like the length and width ratio of the strut of the broken zones. And we also hope to evaluate the impact of broken zone compression on waterproofing capability. So basically, we're gonna try to manually compress the broken zones and see if they maintain their hydrophobic feature. And the ultimate goal of our research is to design optimized broken zone inspired waterproof coating with enhanced durability. For example, can we apply them in a piece of metal and like prevent them from corrosion and rusting? I would like to thank the Chemistry Merit Program for funding this research, the Graduate College at the University of Illinois, the ABC Lab members, the Beckman Institute, the Sandia National Laboratories, and the U.S. Department of Energy. And for the photographs of the St. Insular's and G. Salsa leafhoppers, I would like to thank Alex Ward Photography. Thank you. Here are my references. And thank you so much. And do you have any questions?
Yeah, thank you for that. Well, you already answered one of my questions because I was going to ask sort of what the applications of these kind of, very hydrophobic coatings could be. So you got that one covered.
Thank you.
My second question is a very important one. If you're watching all these Marvel videos, what is your favorite Marvel video?
Ooh, I think mine would be Civil War.
Okay, all right,
Yeah.
all right. Respectful answer. All right. And then I think I'm not sure if maybe you had mentioned this earlier and I maybe just missed it. I know you were talking kind of about the structure of the brachiosomes, but what are they made of? Or like what materials, I guess, are they made of?
So brachiosomes are made of proteins and lipids.
Okay, so more of like kind of carbon-based things.
Yeah.
Okay, all right. Well, I think that's all the questions I have for now. Again, if we have more time at the end, I might have more.
Okay. Thank you so much.
No, thank you. All right. And then last but not least, we have one more presenter for you today. I'm pleased to introduce Viktoriya Zamkova. She is a sophomore working with Professor Jonathan Sweedler and Professor Stanislav Rubikin. And today she's going to share her work on the semi-quantitative cellular and subcellular analysis of protein expression in brain cells using alkaline phosphatase immunohistochemistry. I think I got all of the words correct.
Yes, you did. Okay, let me just share my screen here. Thank you to Dr. Axelson for introducing me. As she mentioned, my name is Viktoriya Zamkova, and I am a specialized chemistry major going into my sophomore year at the University of Illinois. I am part of the Sweedler group, and I was able to do research this summer thanks to the Merit Program Summer Research Scholarship. Today I'll be sharing my research on protein expression in brain cells using alkaline phosphatase-based immunohistochemistry. So first, a little bit about me. I am from Lake in the Hills, Illinois, but I was born and raised in Ukraine, and I moved to the United States when I was 9 years old. I enjoy hiking, playing tennis, and baking. My interest in chemistry initially began in high school when my chemistry teacher told stories of his time as an analytical chemist, and I thought that it sounded super fascinating, so I wanted to learn more. So my senior year of high school, I took AP chemistry, and that's when I really started to see chemistry as more of a career path for me. So, obviously, though, when I got into college, I didn't know for sure that chemistry was what I wanted to do until I actually got to apply it through research. And I think that research has really shown me that I do enjoy chemistry and getting to see all of its different real world applications. So research has shown me that there's still so much to discover and that chemistry continues to allow us to keep learning and exploring the unknown. And I think it's really cool to get to apply what I've learned in classes to advance science and produce new knowledge. So that's something that I very, very much so value. Oh wait, I wanted to also say, so this is a picture of me in one of our labs, and here we have our astrocytes stained with our immunostaining procedure, which I will discuss in the next slide. This is a cake that I did not bake, but I like decorated it and kind of constructed it. This is a picture of me and my best friend, who is also my roommate, at the game day store. And this is a picture of me and my mentor Stas at the summer research symposium. Okay, so moving on to my research. Our goal was to better understand protein expression in specific brain cell types, particularly neurons and astrocytes. Identifying these proteins can help us learn more about how cells function, organize themselves, and interact within the nervous system. And this kind of research can really be applied to neurological diseases and can help us uncover protein level mechanisms to improve diagnostics and treatments. We focused in on two cell-specific biomarkers, the neuron-specific nuclear protein for neurons and the glial fibrillary acidic protein for astrocytes. And we used an alkaline phosphatase-based detection system for the study of rat hippocampal cells. So here we have our experimental workflow. First we have our experimental design development, then we have sample preparation, and then we have our immunostaining procedure. After that, we performed a nuclear counterstain using Hurst, and then we acquired images using phase contrast, fluorescence, and the mix mode. And then we analyzed these images using ImageJ and Zen, and we also evaluated Cell Profiler and QPath, but we didn't end up using those. So our approach is an indirect immunohistochemistry approach, which works by a combination of primary and secondary antibodies. So first we have the primary antibodies, which bind to our target antigens inside the cells. And then we have our secondary antibodies, which bind to the primary antibodies. And these secondary antibodies carry a powerful enzyme called alkaline phosphatase. And when we add our substrate, we see a colorful precipitate, which indicates where our target antigen is and how it is expressed within the cell. So this is our experimental setup, and we have glass slides, which have multi-chambered devices attached to them using silicone. We exposed the first three wells to, these are our experimental wells, we can call them, and we expose them to primary antibodies, secondary antibodies, and our substrate. And then our control well, we only expose to the secondary antibodies and the substrate to rule out non-specific binding. There we go. So when we added our substrate initially, we experienced a lot of background staining because our substrate concentration was too high. So what we did was we attempted to get a time and concentration dependence of our reporter system using time-lapse images and a series of dilutions of the substrate. So here we have a graph of our absorbance versus time course for each of the dilutions, and we have images A to E here showing the increasing red intensity over 20 minutes, and then image F is our control, and it shows no staining, which confirms specificity. And specificity is the ability of our antibody to bind exclusively to its target antigen and not cross-react with any unrelated molecules. And high specificity really ensures that the staining that we observe is representative of the presence and localization of our target antigen. So this graph shows us how absorbance changes over time, and it reflects enzymatic activity. And then this other graph here that we have is our absorbance versus substrate concentration graph, and we tested different substrate concentrations using the same series of dilutions. Images G through J here show how staining intensity increases with concentration. And this is visualized here at the 5-minute mark for each of these images. This graph quantifies absorbance for each concentration and time point, and it helps us to identify optimal conditions for analysis. So what we were really trying to do is identify the optimal concentration for our substrate, so the kind of dilution that we would have to use, and also the optimal incubation time. And then moving on to this middle section here, we have our cell population analysis. So in order to quantify specificity, we analyzed a defined slide region stained with Hurst for nuclei and red alkaline phosphatase for protein expression. And we used image J to count the cells for each of these two channels. And we found that 75% of Hurst labeled cells also showed red alkaline phosphatase staining, which is strong evidence for our target specificity for the glial fibrillary acidic protein expression in astrocytes. Down here in the last section of the results, we have single cell morphometric analysis, and here we examined individual cell morphology. So image T here shows an astrocyte stained with anti-glial fibrillary acidic protein, and its structure closely matches the schematic in image S, which is right above. And this really shows us that this method can help us link protein expression with cellular shape and identity. And then these two here on the right side, we have the RGB signal plots and their corresponding microphotographs. So to further validate our staining, we used RGB signal analysis, and figures U and W here show the intensity of each channel from the images, and the red curve here confirms the presence and localization of the red alkaline phosphatase signal. So moving on to our conclusions. Here we go. So we were able to conclude that immunostaining with alkaline phosphatase reporter system showed time and concentration dependence of the enzymatic reaction product. Our pipeline allows us to investigate heterogeneity of cell populations, cell typing, and morphometric characterization of individual cells. And our protocol is also efficient in that processing can be completed in one workday. In the future, we hope to achieve multiplex immunostaining of the same cells using the same and different color alkaline phosphatase activity products. We also hope to reduce our processing time to 2 to 3 hours so that we can make our protocol even more efficient and we can get more processing done in that time. We also want to evaluate in-house generated primary antibody conjugates with alkaline phosphatase and open up the possibility for direct immunohistochemical analysis. Eventually, though, we would also like to integrate immunostaining, the immunostaining approach with single-cell mass spectrometry so that we can get greater chemical detail for the different molecules within these cells that we are analyzing. So this research was funded by the Neuroproteomics and Neurometabolomics Center on Cell-to-Cell Signaling from the National Institutes of Health and the National Institute on Drug Abuse. I would like to thank my research advisor, Stanislav Rubakin, and my PI, Jonathan Sweedler, as well as the Merit Program Summer Research Scholarship and the Chemistry Merit Program. Thank you.
Well, thank you, Victoria, for that presentation. I did have a follow-up question. I know, I think it was like on that last slide when you were talking about your future directions, you mentioned that you wanted to potentially incorporate some single cell mass spectrometry into the experiment to kind of work that alongside the staining part aspect. what, I guess, like what benefits or kind of what are you hoping to kind of use that marriage of techniques like to study or for?
So cell mass spec allows us to overcome the limitations of the antibody-based methods like immunohistochemistry, which is what we're using. So antibodies are restricted to pre-selected targets,
Mm,
and as I discussed, cross-reactivity may occur. But single-cell mass spec provides an unbiased and more of a large-scale approach that can detect thousands of proteins in
mm,
individual cells. So this really allows us to have more information on the cellular heterogeneity that we didn't have before, and it can give us more of a quantitative view of the proteomic landscape. And this can really help us discover more about neural function um, that the traditional methods can't achieve, like our immunohistochemistry approach. So it really allows us to like expand on what we've been doing.
gotcha. Gotcha. Okay. So I think I haven't seen anything pop up in the Q&A yet. So for any of our attendees, if you have any follow-up questions for any of our presenters, feel free to drop that in the Q&A. In the meantime, though, I do have a little bit of time left. So I guess I had a few more general questions for our presenters rather than, kind of specific technical ones from your presentation. Um, and this can be for any of you or all of you if you would like to take a stab at this. Um, I guess in working, in your research lab and working on your projects, are there any new areas of chemistry or, certain techniques that you've come across that you weren't originally familiar with that have like really sparked your interest or, have caused you to maybe like want to do a deep dive in anything particular that you've encountered so far?
Yeah, for sure. I can answer the question first. So, I think for me, I didn't know anything about immunohistochemistry going into college. I mean, I'd never heard of it. And when my research mentor Stas had first shared the protocol with me, I was reading it and I read glial fibrillary acidic protein and I was like, what is that? I had to like go look it up and, do a little bit of research on it, but now I feel like I know so much more about it and I think it's so interesting. So I think that there's always something you don't know about and something you can discover with research. And I think it's super cool to get to do that.
I can go next. So for me, I worked in an entomology lab, and I didn't really like bugs. I just thought the fact of bioinspiration was cool. And during the summer when we're doing like the insect collecting and everything, like I started to like develop a fascination for bugs. Like anytime I see a bug, I'm like, oh my God, that's a firefly and everything. But yeah, it's a really cool, like you never know what you might like. Just try something new.
Yeah, I kind of had a similar experience where my lab was like really bio heavy, but I hadn't taken like a bio class since like high school. So I had like a lot of learning to do. And approaching biology with sort of like this chemistry mindset, like really opened up how interesting it was for me. Um, and I guess also like one of the first experiments I did was using the fluorimeter. And
Mm,
it seems really simple and it kind of is, but it has like a lot of layers that can go into it. And I remember spending a lot of time just like trying to learn like kind of like the fundamentals. So
mm,
that was really interesting for me.
mm. I think I see, sorry, just looking at participants. Stephen Miller, I see you have your hand up. I'm not sure if we can unmute you to ask your question, or if you want to type it in the chat or in the Q&A, perhaps.
Can you hear me?
Yes, we can hear you.
Yes. I wanted to ask, sorry for coming in late. The last speaker talking about mass spec and electron microscopy, those are great techniques to learn how to use because they can lead to discoveries that you hadn't thought about before. And so I just wanted to check, are these techniques that you actually learn how to do yourself? Because if so, that would be very valuable for you in the future.
Yeah, so I haven't done any mass spec yet, but my research advisor, Stas, he told me that that's something that we want to build onto and like go into. So I think I will get to do mass spec eventually, but right now we're trying to really optimize our immunostaining procedure to really get more information about like single cells and but then we obviously want to do a deeper dive into the actual cell and discover more about the different molecules within them. So he did mention that we would be doing that. Like my lab is entirely based on mass spectrometry. Like they're always trying to optimize the different mass spec methods. So yeah, I think eventually I'll get to do that. So I'm really excited for that.
Yeah, and speaking, I mean, also having been an undergraduate here doing undergraduate research, I know in my personal experience, I mean, it took a little while for me to get my feet underneath me, you know, with my personal research projects and things. But overall, I felt that like my mentor also did a really good job of like letting me go and like use the NMRs when that became, you know, kind of an integral part to like my projects and being able to analyze what I was making in the lab. So, yeah, I think in general, I feel like our faculty do a pretty good job of, you know, as the student becomes more, you know, independent and comfortable in the lab, that, you know, they're allowed to delve into the experiments that, you know, just correspond or complement their projects. And I guess I'll have one more question. Just kind of thinking about either, you know, the work that you've done over the summer, or, you know, for those of you who've been in the lab a little bit longer, um, of all the things that you've done or like learned or accomplished, what have you been most excited about, or like what are you most proud of accomplishing so far?
I guess I can go first. Um, I guess the biggest thing or I guess like the thing I'm most proud of is like my growth. Like starting in the lab, I was going into my junior year, and at that time I had only taken like lab classes, so I didn't have a ton of experience. And compared to then, I was just like following my grad mentor. He would like conduct or he would like design all the experiments, and I would just do them. And then now I'm like starting to design my own projects, design my own experiments day to day, and I think that's the thing I'm most proud of.
I can go next. So I think for me, there's two things. One is being able to do the experiments independently because in the beginning I was really scared to mess things up because I didn't know how to do any of our like experiments. I mean, I didn't even know how to use one of the micropipettes, like with where you had to draw out like a very small volume. So I was really scared that I would just mess it up. And I think one of the most important things that my research mentor taught me, Stas, is that it's okay to like mess up, but you just have to keep going. You have to keep trying and like eventually you'll get it right, you know? So I think that's one of the things, being able to do the experiments independently. And the first time I ever did like the experiment on my own from start to finish, I was so happy. I called my parents. I was like, oh my God, this is so exciting. And this was actually over the summer. So I thought it was really cool. And also one of the other things, I had another thing, but I'm kind of blanking now. So I might come back to it.
For me, I have a lot of things that I'm really proud of. Well, I'm genuinely proud of like my growth and being enthusiastic in general. Like I worked in the lab also during the spring semester, and my grad mentor like taught me like led me through the process of how to use the scanning electron microscope at Beckman. And not gonna lie, I was kind of rusty. And when I came back during the summer, and she also guided me, and I think like after three more trainings, I was able to go there and start taking pictures myself, and I was so proud of myself. And also I've basically like been like curious. I'm like, oh my God, what are we gonna do next? And I'm just excited to continue in the fall too. And yeah.
Well, I think that is all the questions that I have for today. Again, I don't see any more in the chat or anything like that, so I think we're good to go. So I guess I will kick it back to Amanda to finish us up.
I wanted to say thank you to everyone who joined us today. Please join me in thanking our speakers for their time. We hope you enjoyed this event, and please keep an eye out for our future alumni virtual events. In the meantime, we wish everyone a great rest of your day, and thank you again for coming. Great job, everyone.